హలో హలో హలో
హలో హలో
హలో హలో సరిపోద్దుగా ఇది ఎలాగా లౌడ్గానే మాట్లాడనా కావాలి మీరు అడ్జస్ట్మెంట్ ఉంటే మా అక్కడ చేసేస్తా ఆయన పెట్టేసి distinguished professors, guests, faculty members from various colleges, scholars, and student participants. A very good afternoon to you all and a warm welcome to you. It's a privilege to stand before you today as we gather for the national seminar on aquaculture farming, advances, challenges, and opportunities. This event marks a significant milestone in our collective efforts to explore, understand, and promote the growth of aquaculture, an industry that plays a vital role in ensuring food security, economic development, and environmental sustainability. I am confident that the next few hours will be filled with enriching discussions and exchange of ideas that will propel the aquaculture industry to new heights. Ladies and gentlemen, please join me in extending a warm welcome to our principal and president of today's event, Dr. T. Rajarajaswari Garu, to grace the dais. I request Dr. M. Ramakrishna, organizing secretary of today's seminar, to honor principal madam with a plant. With a great pleasure, I invite Professor C.H. Manjulata Madam, Chairperson BOS, Department of Zoology, Andhra University, to join us on the dais. Her wealth of knowledge and expertise will undoubtedly enrich our seminar proceedings. I call upon Dr. V. Yamini Madam, Department of Chemistry, to present her with a plant. Photo. I would like you to join me in welcoming a man of profound intellect, Professor G. Simhachalam, Department of Zoology and Aquaculture, Acharya Nagarjuna University, to come on to the dais. I call upon our IQC coordinator, Sri Bhadrachalam sir, to present a plant. We are privileged to welcome Dr. A. Mattareddy, who doesn't need any introduction. He is a jewel among academic fraternity of aquaculture. I call upon Dr. C.H. Usharani Madam to present a plant to sir. Now I invite Sri T. Krishna, Vice Principal and COE of Sri A.S.N.M Government College, to come out to the dais and occupy his seat. I call upon Srinivas, sir of Botany Department, to present sir with a plant. I invite Sri K. Badrajam sir, IQAC coordinator, Sri A.S.N.M Government College, to come over to the dais and occupy his seat. I call upon Dr. D.V. Lakshmi madam to present sir with a plant. Now I invite Dr. M. Ramakrishna, organizer secretary of today's seminar, to come over to the dais and occupy his seat. I call upon Sri Sivakrishna, Department of Mathematics, to present sir with a plant. Now let's begin this seminar with a prayer to center ourselves and foster a collective sense of reflection. I invite Kumari A. Jansi, Kumari D. Tulsi, Kumari Haima Durga Bhavani, and Kumari P. Shravani of 3rd BSc, CBZ and HBC for prayer. I request all to stand up for the prayer.
या वीणां वरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युत शङ्करप्रभृतिभिः देवैः सदा पूजिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापः निःशेषजाड्यापः.
Thank you. I invite our esteemed guests and dignitaries on the dais to join us for the lighting of the lamp ceremony, symbolizing the illumination of knowledge. I request our botany sir, Srinivas Garu, to look after this. Thank you all. So without any further ado, I invite our principal madam, Dr. T. Rajrajeshwari, to share her insightful remarks guiding us through the seminar.
Good morning to all of you. Respected Chief Guest, Dr. David Kumaraswamy, Joint Director of Collegiate Education, Mangalagiri, distinguished guests of honor, faculty members, scholars, diligent students, and distinguished participants, I extend a warm welcome to each one of you to our esteemed institution as we host this significant national seminar on aquaculture farming advances, challenges, and opportunities. It is indeed an honor to have experts, scholars, enthusiasts gather here to delve into critical facets of aquaculture, a topic of immense relevance to our evolving world. At the outset, let me express my appreciation for the organizing secretary, Dr. M. Ramakrishna, in charge of zoology and aquaculture, guest faculties of the Department of Zoology, Ms. Josna Kumari and Ms. Hepsi Rani, Sri T. Krishna, Vice Principal, Lecturer in Political Science, Sri K. Bhadrachalam, IKC Coordinator, Lecturer in Computer Science, and members of the various committees of the seminar who have worked tirelessly to make this seminar a reality. I express my warm wishes and thanks for accepting our request to act as a resource persons for today's national seminar. Sri Dr. A. Mattareddy Garu, Associate Professor of Adikavi Nannaya University, Rajamahendravaram, Professor Manjula Chapara, Andhra University, Department of Zoology, Visakhapatnam, Professor Simhachalam Garu, Acharya Nagarjuna University, Guntur, Sri A. Venkateswarrao Garu, Environmental Engineer, Regional Office of AP Pollution Control Board, Yeluru, and participants from various colleges are the academic pursuits, and they are integral to the ethos of our institution, where knowledge is not only important but also actively created and disseminated. When we look into the advances in aquaculture farming, aquaculture has witnessed remarkable advances in recent years, revolutionizing the way we cultivate aquatic organisms. Technological innovations, research breakthroughs, and sustainable practices have propelled the industry forward. Precision aquaculture, that means harness the power of data, analytics, and automation to optimize feeding, monitor water quality, and enhance overall productivity. Selective breeding techniques have enabled the development of resilient and high-yielding aquatic species, contributing to increased efficiency and reduced environmental impact. Our academic community has played a crucial role in contributing to these advancements. Today, we have the opportunity to celebrate the progress achieved and explore the avenues that lie ahead. The challenges facing in the aquaculture industry are, however, as we celebrate these achievements, it is crucial to acknowledge the challenges that loom over the aquaculture landscape. Environmental concerns, disease outbreaks, and the delicate balance of aquatic ecosystems demand our vigilant attention. Overfishing and habitat degradation pose threats to the very resources upon which aquaculture lies. Addressing these challenges requires a holistic approach, integrating science, policy, and responsible business practices. When we come to discuss about the opportunities for sustainable growth in aquaculture, in the face of challenges, opportunities emerge. Aquaculture stands as a beacon of hope for meeting the escalating global demand for seafood while alleviating pressure on wild fishes. Sustainable practices such as integrated multi-tropic aquaculture and recirculating aquaculture systems offer environmentally friendly alternatives. Collaboration between governments, industries, and research institutions can foster the development of policies and technologies that ensure the responsible growth of the aquaculture. Now it is the need to empower the local communities. Aquaculture is not just a global industry, it is a lifetime for countless communities around the world. By empowering the local fish farmers with knowledge, resources, and access to markets, we can promote economic development and food security. Small-scale aquaculture enterprises, when supported and guided, have the potential to become catalysts for positive change, lifting communities out of poverty and contributing to sustainable development goals. For this national seminar, if we think about fostering of academic dialogue, this seminar provides a platform for fostering academic dialogue, collaboration, and the exchange of ideas. I encourage our students to actively participate and engage with the distinguished speakers, seize this opportunity to expand their knowledge base beyond the classrooms. Encouraging future leaders. To our students, I say, you are the future leaders and innovators in the realm of aquaculture. The challenges we face today are the opportunities for you to shape a more sustainable tomorrow. I urge you to absorb the insights shared during this seminar, ask questions, and let your curiosity fuel your academic journey. A vision for sustainable aquaculture. As we embark on this collective exploration, let us envision a future where aquaculture not only meets the nutritional needs of growing population, but does so in a manner that is environmentally conscious, economically viable, and socially responsible. Before closing my remarks, I express my heartfelt thanks and gratitude to my friend and adjacent roommate, in Research Scholars Hostel and also in Girls Hostel of Andhra University. She is my senior. After 1992, just now I am seeing her, Dr. Professor Manjula, Madam. She is staying in just to my adjacent room in Scholars Hostel of Andhra University near Siripuram. When I have to get her phone number from somebody, and when I called her, she accepted my invitation and my request. I am eagerly want to see you. We want to talk with each other and discuss about our staying in hostel, our studies, everything. I am very much thank you, Manjula, for accepting our invitation. She is so simple. Today, she came to Bhimavaram railway station. I told her, we will send the car, but she does not like. I am very simple, environmentally friendly. Why we should waste the petrol for these 20 kilometers? I always likes only the public transport, she said. So she waited. She get down from the train by 3:30, and she waited till 6 o'clock. And the train came from Narsapuram to Bhimavaram. Then I called her around 6:15. She said, I catched the Narsapuram train. Now after reaching Palakollu, I will come there. And we want to, because she is the professor, she is the senior most professor in Andhra University. The seniors are already retired. So we want to accommodate in some restaurant, but she does not like that also. So as a lady member, so in reception and in accommodation, we approached Padmalata madam. She said, Madam, I will take her to my house. So thank you, Padmalata madam, for encouraging our guest to this and your stay in our house and your hospitality. Thank you from the college and from the staff. Thank you, Manjulata madam. She said, if you are like that, then many more times I will come to the college to disseminate my knowledge, and if it is useful to the students and the society, this is the only my motto, she said. She had one daughter. She completed her MBBS. Now she is preparing for PG medicine. In the March, they are going to write. So this is about my friend Manjula. And also now, Simhachalam sir, I approached him through my Professor Ramachandra Rao sir in Department of Chemistry, Nagarjuna University. He said in aquaculture, Simhachalam sir is there. Then after contacting him, he accepted my request. Thank you, sir. Simhachalam sir, thank you very much for accepting my request. And Matareddy sir, I know him. He is also very simple and rigid. They are all the stage resource persons. They are all very simple and strict to the to their discipline. Their strictness shows their discipline. They won't bother about anything. Only their ambition is to tell something to the students. So thank you very much, sir, Matareddy garu. Because before coming to this place also, I had relation, because we both went for inquiry to one college 2-3 years when I working as a principal in Neluru College. So thank you very, sir. And my heartfelt thanks to Dr. M. Ramakrishna sir, because I told him, because you are a doctorate, so you have to apply for aquaculture. Because when I approached AP Pollution Control Board for during the purpose of NAC for green initiatives and energy initiatives, that A. Venkateswara Rao garu, who is coming in the afternoon, he said, Madam, you conduct one seminar, because in West Godavari mostly aquaculture field is there. So the impacts, the pesticides they are using and their effects, we want to explore our ideas and our thoughts to the students, so that it is very useful to the local people also. And he said that we will give financial support also. Then during the NAC time in September 2022 onwards, I am telling him, after NAC, then we are a little bit, even though we are now little bit busy with our submission of AQR 2022-23, but then he never says no. When I said, sir, you do this one, then he always obliges my request. So thank you, sir. And my staff, Vice Principal sir and IKC coordinator sir and other staff who are involved in the culture, in these committees, they, because we are all new to this college, the staff and myself also new to this college, so they are all eagerly waiting to celebrate this type of national seminars and to take this college to great heights, because in the research criteria 3 we are having this type of seminars, conferences. For that we had the marks, so we are planning. In conclusion, I am closing. I extend my gratitude to our distinguished guests, eminent speakers and all participants. May this seminar be a source of inspiration, learning and collaboration, paving the way for a more sustainable and thriving future in aquaculture. Thank you and wish you all fruitful deliberations throughout the seminar. Thank you.
Thank you so much, madam, for your beautiful and inspiring words about our keynote speakers today, covering a large aspect of our entire seminar. I'm very much thankful to you from all the participants and the students who attended the seminar. Now I invite Dr. M. Ramakrishna, Organizing Secretary of today's seminar, to elucidate the seminar theme and provide elucidate the seminar theme. Thank you.
Respected our beloved principal, Dr. T. Rajarajeswar Garu, as chairperson of this national seminar, and all the dignitaries already our beloved madam introduced various prestigious universities, came from various prestigious universities to here. So I don't want to again introducing, though because of the time is very much time is enough. So I am directly entering to my topic. This one is the theme of this national seminar. Before that, various colleges lecturers also came here. I thanks to one and all. First of all, the Department of Zoology was established in the year of 1968 here. So on that one, our department conducted, this one is, I think this one is the second seminar, national seminar. First one, Dr. M. Vijay Kumar, one national seminar was conducted. Now we are conducting this one. Actually, the theme of this seminar is technological advancements, sustainability and environmental impact, aquaculture in a climate, changing climate, and lastly, processing of aqua catch in environmental friendly manner. These four are the main themes to deliver our various professors. So this is the main important topics covered in the aquaculture farming, advances, challenges and opportunities. Now I am very much glad to our professors, because immediately they are accepted my request They came very long distance. So I am once again thanks to one and all the professors, and also thank you one and all. Please be carefully listen and be maintain a discipline. Thank you one and all.
Thank you, sir. Now I request our chief guest and other distinguished guests to kindly join us for the ceremonial release of souvenir. I request souvenir committee to come over and continue the processing. I request all the dignitaries to join in the release of souvenir. Thank you. Now it's an honor to invite our distinguished chief guest, Dr. C.H. Manjulata Madam, to enlighten us with her valuable perspectives on the seminar. I request our distinguished chief guest to enlighten us with our valuable perspectives on the seminar.
Okay, good morning all of you. Afternoon, sorry. Good afternoon all of you. So it's a very wonderful day for all of us, because it's a very rare conducting such, not a very easy task for conducting a seminar. But my dear friend Rajarajeshwari, she's achieving so many laurels here. So now I wish this seminar also will help the students particularly. This is aquaculture is a very important topic nowadays worldwide. So this is very important for particularly the students. So the main thing how the people students will be get advantage from this seminar. So through aquaculture, so you can have lot of knowledge first of all, and also so it's a very important employment field. So you can come and you can become the entrepreneurs also with this. After having this knowledge, you can start on your own, and you can employ some more people. So I hope this definitely this will help the students particularly for this college. Thank you.
Thank you so much, madam. Now I request our Professor G.C. Mahachalam sir to address the gathering for 2 minutes.
I congratulate the organizers behind the national seminar, and I wish to great success this national seminar. Thank you very much, and thank you.
Now I request Dr. A. Mattareddy sir to address the gathering.
Respected Principal Garu, Rajarajeshwari Garu, respected Professors Manjulata Garu, Simhachalam Garu, and Head of the Department Ram Krishna Garu, Vice Principal Krishna Garu, other faculty members and faculty members from various colleges, and my dear students. This aquaculture farming challenges, advances, opportunities is a unique theme, because this region is very rich in aquaculture. As experts, we need to address the problems of this region. If we meet in this manner in various seminars, conferences, we can discuss the problems of the aquaculture industry of this region. We can help at least in some way or other way, because there are a lot of problems faced by the aquaculture industry, and all of us should rise up to the occasion, should work harder, train the students, and supply the human resources to the industry. Just technical knowledge means it is not only water analysis. Over and above the basic techniques, we need to give the various tools and techniques in genetics, genomics, proteomics, gene editing, etc. That is the reason why Dr. Ramakrishna Garu included several themes ranging from normal farming techniques, improvement, crop improvement, and how to enhance the yield by various culture techniques, as well as making an efficient research at gene and protein levels. So definitely, I wish all the people will learn something out of these lectures, be enlightened, at least, especially I wish all the students should pay attention for the lectures and learn something from the experts attending here. Thank you very much.
Thank you, sir. Okay. Now I request our Vice Principal, sir, to wish the gathering.
అందరికీ నమస్కారం. వేదికలు అంకించిన పెద్దలందరికీ నమస్తే. అలాగే ఈరోజు నేషనల్ సెమినార్ జువాలజీ డిపార్ట్మెంట్ తరఫున జరుగుతున్నందుకు చాలా సంతోషిస్తూ, అలాగే మరి మేము ఆర్ట్స్ సబ్జెక్టు వాళ్ళం, ఈ సబ్జెక్టు మీద కొంచెం అవగాహన తక్కువగా ఉంది కాబట్టి వాళ్ళు ఎవరైతే సబ్జెక్ట్ ఎక్స్పర్ట్స్ ఉన్నారో వాళ్ళ వినేసేసి స్కాలర్స్ మరియు స్టూడెంట్స్ దాని తగ్గట్టుగా ఉండవలసిందిగా కోరుతున్నాం. థ్యాంక్ యూ.
Thank you, sir. Now I request our IQAC coordinator, Sri K. Bhadrachalam, sir, to address the gathering.
Respected Principal Madam Garu of this college, all the dignitaries on the dais, my dear faculty members and students, good afternoon everyone. This is K. Bhadrachalam, working as a lecturer in computer science and IQAC coordinator of this college. As a part of internal quality of the institution, Department of Geology come forwarded and organized this type of event. Firstly, I congratulate Dr. Ramakrishna Garu and his faculties of Geology Department. And I thank Principal Madam Garu for her support and encouragement for conduction of this event. And actually, we have been doing a lot of activities from 2 years onwards, but we didn't conduct any events like this because majorly we have been doing work related to NAC. So recently we faced NAC and we got some better grade, and even we are in busy situation, so we try to submit, we are in the final stage of submission of AQR also, so even we are having examinations. So along with all these works, so Department of Geology conducted this type of event, right? So for each and every participant who are here, so I thank with your support, so we are going to conduct many type of events in future. Thank you everyone.
Thank you, sir. So, for our national seminar, we have chosen 4 eminent speakers. So, we are about to start our technical sessions. So, we have 2 technical sessions. In the first session, we have 2 speakers. And after that completion of our lunch, we will continue with the 3rd and 4th keynote addresses. Now I invite Josna Sri, Department of Geology, to introduce our first keynote presenter for the session 1.
Good afternoon, all of you. I extend my warm welcome to the one-day national seminar on aquaculture farming, advances, challenges, and opportunities. I am delighted to introduce our distinguished resource person, Dr. A. Mattareddy sir. Dr. A. Mattareddy sir is an eminent academician and a revered figure in the field of zoology, currently serving as a distinguished professor in the Department of Zoology at Adikavi Nannaya University, Rajamahendravaram. With a career spanning several decades, Dr. Reddy had made significant contributions to the realm of zoological researches, earning him recognition and respect both nationally and internationally. His academic journey is marked by an impressive educational background and a passion for unrelieving the mysteries of the animal kingdom. Dr. Reddy holds advanced degrees in zoology, attesting to his scholarly prowess. His research interests encompass a wide array of topics within the discipline, ranging from biodiversity conservation to behavioral ecology, showcasing the breadth of his expertise. Dr. Reddy's has commitment to excellence extends beyond the confines of his research lab. As an educator, he played a pivotal role in shaping the next generation of zoologists. His teaching philosophy emphasizes a holistic approach to understanding the intricacies of natural world, inspiring countless students to pursue careers in biological sciences. Beyond his academic responsibilities, Dr. A. Maddareddy has actively participated in various scientific forums, presenting his research findings and engaging in collaborative efforts to address pressing issues in zoology. He was a recipient of Excellent Presentation Award in KSSCR 2006 in Korea. As we welcome Dr. Reddy to the National Seminar on Aquaculture Farming Advances, Challenges and Opportunities, we anticipate insightful contributions that will enrich the discourse on contemporary issues in aquaculture. His presence is sure to evaluate the intellectual exchange and inspire fellow researchers, making this seminar a platform for valuable insights and advancements in this field. Without further delay, I invite Dr. A. Maddareddy sir to take the stage and share their keynote presentations with our audience. Thank you.
Good afternoon, everybody. So today's my lecture is the use of CRISPR-Cas gene editing technology in aquaculture. CRISPR-Cas technology is very young field. This was first done in 2013. Actually, CRISPR-Cas sequences were discovered long back, almost 3 to 4 decades ago. Those sequences were identified, but in the laboratory, in the animal cells, gene editing by using CRISPR-Cas was done for the first time in 2018 in animal cells, and in the same year, later months, in the plants also, CRISPR-Cas technology was used to edit the genes. So editing genes is a fascinating area of research because it has both positive and negative sides. One should be very careful while editing the genes. The gene editing need a lot of expertise. Otherwise, it may cause very harmful effects, and the escapees may induce a lot of problems in the nature. Escapees means if you are doing this type of gene editing experiments in controlled conditions, it's okay. In laboratory conditions, sometimes if it is failed and if the organisms escapes into the nature, again they may breed with the normal animals and they may cause a lot of problems. So that's why while conducting these experiments, one should be very careful, and we should use this technology with utmost care. So let us have an introduction to the genome editing. So genome editing is necessary to address several problems. Sometimes if mutations or genetic errors, if they are present, they should be corrected. And sometimes to avoid pathogenesis, to avoid infections, we need to edit the genome. So there are different purposes. Gene editing has a lot of purposes. So this is these are different types of gene editing techniques. The first one is zinc finger enzymes. Zinc finger protein acting as enzymes, which will be cutting the sequences. The double strand breaks may be induced with the zinc finger protein enzymes. So these enzymes are acting as nucleases. Most of you know about the nucleases. Nucleic acid breaking enzymes are called nucleases. Usually DNA breaking enzymes are mainly of two types, exonucleases and endonucleases. So let us have some basics about the nucleases, because most of my lecture is dealing with the nucleases, different types of nucleases. The basic nucleases you might have already known in your lower classes are endonucleases and exonucleases. Exonucleases will be cutting the DNA nucleotides from the tips, from the terminal points. Exonucleases cut nucleotides every time one nucleotide from the tips on either sides. So that is a duty, that is a function of exonuclease. Whereas endonucleases can cut in the middle of the sequences, but they cannot cut at any place. They cannot cut invariably or randomly, they cannot cut. Endonucleases have restriction sites. Only at specific sequences these endonuclease enzymes will be cutting. That means each and every endonuclease type is having a specific site of cutting. It recognizes the sequence, for example, AAGGCC. Wherever AAGGCC is there, it will be cut down. Like GCTA, wherever GCTA is there, it will be, another enzyme will be cutting. Like EcoR1, HIND3, there are several restriction enzymes. They have their own recognition sequences. Only whenever they find these sequences, they will be cutting the DNAs. So this is about nucleases. So there are different types of nucleases, like endonucleases. These are also the other type of nucleases, zinc finger protein nucleases, and again the other type, TALEN. TALEN also can cut the sequences at different places, specific sites only. And this is the CRISPR-Cas, what we are going to talk for the next 30 minutes on the CRISPR-Cas. So CRISPR-Cas is a long abbreviation, clustered regularly interspaced short palindromic repeats. Cas. Cas means CRISPR associated nucleases. They are actually proteins, and those proteins acting as nucleases. So in full form we can call CRISPR-Cas. CRISPR-Cas means clustered regularly interspaced short palindromic repeats. I think most of the students are biology students, right? Most of you are from biology. So I will make it simple, because the abbreviation looks very complex and the meaning appears very complex, but I will make it in a simple way. Don't try to by heart the abbreviation. I will tell a small technique to remember this abbreviation. Come from the last three letters, short palindromic repeats. Short palindromic repeats, you know very well in DNA. Short sequences which are palindromes. palindromic words you know very well, like Malayalam. From either sides, it is Malayalam. So palindromic sequences are also having repetitive sequences from either sides on two strands. Not the same sequence on the same strand is repeating on either sides. It is present on two opposite strands and having repetitions. So those are called palindromic DNA sequences. These palindromic short repeats, repeating again and again and again. That's why they are called short sequences, and they are palindromes, and they are repeating again and again and again. So this is about short palindromic repeats. And the first three letters, CRIS, CRI. CRI means clustered, regularly interspaced. These short palindromic repeats are in group. That means some 15, 20, 200, 250, 300 sequences repeating again and again. So these clustered, regularly interspaced, they have a gap. Gap means other sequence between these repeats, just like AAGGCC, 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 AAGGCC. In this way, 200 times. But they are interrupted by other sequences in very regular manner. For example, 6 nucleotides, a gap, maybe with 10 nucleotides. Again 6, 10, 6, 10, 6, 10. So this 6 nucleotide short palindromic repeat is again and again repeating with the same space, same interval, same number of nucleotides, random nucleotides. They are not systematically arranged nucleotides. The gap sequences are having random nucleotides, but these repeats are the same nucleotides 200 times, 300 times they are repeating. So what is the speciality of these repeats? Clustered, regularly interspaced, short palindromic repeats. These repeats have lot of specialities, lot of importance in the genetics and molecular biology. The variations among individuals also is with the number of repeats at different chromosomes at different sites. The variations among our phenotype may be because of this type of numbers different with different people. And these repeats are with different numbers in different individuals. For example, in a particular area, these short palindromic repeats may be 106 in chromosome 1 in one place, in me. And in some X, it may be 108. In some Y, it may be 120. And in some Z, it may be 96. That means, based on these number of repetitions, you can identify people. That is DNA fingerprinting technology. You might have already studied DNA fingerprinting technology. So, number of repeats are different with different individuals, like your fingerprint, how it is different among so many people. The repeats are also different. That means the repeat sequence is not different. The number of repeats are different. And I think you got my point here. For example, the repeat sequence is GC80, GC80. The same GC80, GC80, GC80 will be repeating several times. In one fellow 100 times, other fellow 106, the other fellow 110, and in another fellow 150 times. It may be repeating. So that is a unique signature or fingerprint of each and every individual. That is VNTR, variable number of tandem repeats. For identifying the people by seeing their sequencing, we can identify. Even the relationships between the family members, among the family members, daughters, sons, parents, brothers, sisters, we have relationships with these tandem repeat numbers. Again, that's a different subject. If tandem repeat number exceeds some number, that's a problem. If tandem repeat number, if it is less than minimum number, that is another disease, genetic disorder or disease. So there is a range for every repeat, how many times it should be present among human beings with different populations. So the same number, as long as we are having the repeat numbers falling in the normal range, we will be normal. If our repeat number exceeds the normal range, we may be abnormal with the genetic disorders. Or less than the minimum number, again we may have genetic disorders. So now we come back here. These repeats are very important in gene editing, and they are easily be managed by creating cutting sites. That means here by using these repeat sites, you can cut, you can edit DNA at whatever place you want by using the enzymes called nuclease enzymes. So here we target a particular sequence. We target a particular sequence by sending an artificial oligonucleotide into the organism or the cell or any plant or animal. Then this will go and identify and bind the complementary sequence. For example, G sequence is there. If you want to target, what kind of probe you need to select? Opposite. C. Complementary strand you will select and attach nuclease enzyme to that, send into that, it will cut at that particular place. It will go and bind, identify, and then cut at that place. After cutting, again joining, recombination is always present in the organisms. The recombination will provide joining, end joining. If it is DSB, double strand break, one type of recombination. If it is single strand break, that is easier for again joining back. So by recombination, non-homologous end joining, or by homologous end joining, by following various types of recombination methods, these ends will be joined together. So this is a brief introduction about the CRISPR-Cas. Yeah, I don't want to go into details of this because these are different gene editing technologies available until now, and these two technologies, zinc finger protein nucleases, and again these two tunnel technologies, these two are old technologies. Until 2013, these two technologies were used, and after 2013, CRISPR-Cas. So this is a new technology, only 10 years old technology. Still we don't know many things about this CRISPR-Cas technology, and commercially it will take a little bit more time to use this technology, because you have to study about in and around of this technology a lot. See this one, this is a repeat, a gap, that means a normal sequence, and again a repeat. So this is a repeat, So the repetitive sequences are having an interval, a regular interval. In this way, these repeats will be coming and again and again. After processing, a hairpin loop is produced, and this loop can be used for gene editing. Actually, this is naturally existing in bacteria. You know very well, we have immune system. Any bacteria or any foreign cell enters into the body, our immune cells will take care of, and they will manage to keep our health in good condition. But in bacteria, it is a single cell. How it is managing? Doesn't they have any enemies? They have some enemies like viruses. Viruses may infect bacteria. They may kill bacteria. So how to protect from the viruses? They have this technology. Actually, these CRISPR-Cas sequences and CRISPR-Cas editing, this was discovered in bacteria, where they are naturally using this technology to remember the virus sequences, because virus sequence will be incorporated into the chromosomal sequence of bacteria, and for the next time, if the same type of virus visits the bacterial cell, this nuclease will be cutting the viral DNA and dismantling viral DNA. So this is the way how it is having a defense system. So from this, we have learned this CRISPR-Cas gene editing technology, and we are now trying to use this technology for controlling diseases in animals, controlling diseases in the plants. Even in future, we may develop technology to control diseases in human beings also. This is very early to say we can use this technology even for human beings, but definitely in the coming decade of period, we can use this technology for human clinical trials also. So this is about the history of the CRISPR-Cas technology. Even though they were first identified in E. coli in the 80s, and further research happened, and finally it was this technology was used in animal cells in 2013, and the same year this was used in the plant cells also in the same year. Then how does CRISPR working? There are two sequences here. If I show you the picture, that will be easier to understand rather than understanding this matter. Just remember here the DNA, RNA, and adjacent sequences. These three are playing important role here, along with a protein. That's why these complexes are called DNA, RNA, protein. RNA plus protein is used as a therapeutic molecule. That's why they are called RNP, ribonucleoproteins. And spacers and protospacer adjacent motif means a sequence again. Yeah, this is the mechanism of bacterial defense system. If virus sends the DNA of virus into the bacteria, it will be incorporated into the bacterial chromosome, and then it will be remembering, and next time if again the same type of viral DNA comes into the bacteria, it will be dismantled by using this type of CRISPR-Cas proteins. That means they are nucleases. They are powerful nucleases, they can dismantle the viral DNA. So these are the words we need to remember here, vocabulary. They are the protospacers, that is DNA. Protospacer means DNA, and spacer is RNA. RNA is called spacer, and then protospacer adjacent motif. Protospace adjacent motif means in the DNA sequence, ఈ protospace పక్కనే ఉండే sequence. Motif is a sequence pattern here. These three words you remember here. Yeah, see this one, this figure. This is the DNA, double strand DNA. Double strand DNA opened up at this particular site, and this one, this blue to yellow, this sequence we have introduced into the cell. So this is CRRNA, that means CRISPR RNA. CRISPR sequences produces RNA by transcription. So this is CRISPR RNA, and this is the spacer sequence, which has a complementary sequence to that of DNA sequence. ఇక్కడ ఏముందో దీనికి ఆపోజిట్ సీక్వెన్స్ ఉంటే బైండ్ అవుతుంది. బైండ్ అయిన తర్వాత this CRISPR RNA is carrying tra-CRRNA, that means transactivating CRISPR RNA. When it is made sure ఇక్కడ బైండ్ అయిందనే గ్యారెంటీగా అష్యూరెన్స్ వచ్చిన తర్వాత, then this starts its action and acts as molecular scissors and cut down this DNA at two places. That means DSB, double strand break. So this is actual process of gene editing by using CRISPR-Cas. This is the enzyme, this is the protein acting as enzyme, and this is the RNA. RNA plus protein are joined together. This is called RNP, ribonucleoprotein complex. That means RNA plus protein, and this complex, when you introduce into a cell, this identifies the particular targeted sequence based on this sequence. This is oligo RNA sequence you designed and send into the cell. Once this is bound with the DNA, this acts, and this is promoting this enzyme to cut at these two sites, and the DNA is broken into two pieces. This is the result. Yeah, after cutting of the DNA, again the cell has its own mechanism to join them back together by homologous joining or non-homologous end joining procedures. It may have a joining procedure. Sometimes some sequences will be deleted, it becomes short, and sometimes new sequences can be introduced by using this technology. A new sequence can be made, and a long sequence can be introduced in between these two fragments, and the length of the sequence can be increased. So these are all different possibilities available in your hand. And this is again how to introduce the HDR for gene edition, homology directed repair, HDR. And this is normal repair process, and in this way also you can introduce new sequences of your interest into the DNA. That means if you want to enhance the yield, or if you want to introduce new disease resistant genes at a particular place, you can do this. And then CRISPR genome editing workflow follows with the first you have to clone your Cas enzyme, choose and clone, make or synthesize your Cas enzyme, and then you have to make a guide RNA. You can make a guide RNA, you can order. If you know the sequence of a particular organism, then you can understand where you can target. Here is a question: how to know about the sequence of the organisms? These days, whole genomic DNA sequence doesn't cost much. It is less than $10. Even you can have your own genomic DNA sequenced and stored for in future use. So it is less than $10 to get genomic sequence. Already more than 70 aquatic organisms were done with the genomic DNA sequencing. In future, even hundreds of aquatic animals can be done with genomic DNA sequencing. All the genomic DNA sequences completed will be available in the NCBI GenBank, and from that GenBank you can download the sequence and choose where you can target a particular sequence. What do you want exactly, whether you want to cut down and remove some sequence or introduce a new sequence, or one point mutation you would like to introduce, depending on your requirement. First you should have a design, a strategy, a plan. You have to decide first. This is only a technology, this is only a tool. For example, if you find a scissors, you can use scissors for so many purposes. So this is also like a scissoring technology, cutting technology. By using this, again you can introduce the desired sequences at desired places, for which you need these guide RNAs, and then deliver the RNPs, RNA plus protein complex. You have to deliver to the right place where it has to do work, to do function, and then repair whatever you want to repair a particular sequence. You can do it, and finally after repair you have to analyze whether your repair is successful or failed. If it is failed, again you have to conduct the experiment back next time. So analysis may be again by having sequencing, DNA sequencing, by using NGS. And then there are several applications for this CRISPR-Cas technology. You can make a screening of various genes present in an organism, and also you can make CRISPR cutting without double strand breaks also. It is not always just cutting two strands in opposite strands at same place. Without cutting double strand breaks, single strand break also, sometimes you can introduce new sequences. And also you can make gene silencing. Gene silencing can be done by using dead Cas9. There are two enzymes, Cas9 and Cas12. These two are attached. That means, depending on the experiment, Cas9 is very old. For the past 10 years it is being used. Cas12 is a little bit more recent one. But Cas9 was popularly used to cut the DNA sequences. Depending on your need, you can choose Cas9 or Cas12. But here, Cas9 is made invalid that is dead Cas9. That means it don't cut the DNA. It don't cut the DNA. Still you can use that Cas9 protein in this by using this CRISPR-Cas technology. How you can use? This will be going and binding the DNA and keep quiet. That means it is not allowing other mechanisms, other enzymes to come and join with this DNA, and that gene will be made silent. This is called gene silencing. So overexpressing genes can be made silent by using a dead Cas9 enzyme, which can be attached to guide RNA by using CRISPR-Cas technology. It will be going, but Cas9 is not active. That is, nuclease enzyme is not active. It is inactive, but still it is going and binding the DNA, arresting all the DNA expressions. So this silencing is very much useful in the overexpressing genes like oncogenes, cancer genes. Cancer genes will be producing cancer proteins. As long as cancer proteins are produced more, the tumor size will be increasing. You can arrest such oncogenes by sending this type of dead Cas9. And then gene activation you can make by using trans-activating sequences, and again other non-coding functions with the dead Cas9 containing ribonucleoproteins, and nuclear organization and epigenetic modifications also possible. It is not always expression of a gene or silence of a gene is making big difference. Sometimes in the nucleus, chromosomes are folded and bound by proteins, and they are not allowing enzymes to continue transcription, because the organization of the chromosome is not allowing the starting of gene expression. That means the transcription cannot be started. So such problems also can be solved by using this CRISPR-Cas technology. Okay, until now we try to understand the CRISPR-Cas technology, how and where we can use in the fish biology, aquaculture, how can we use. I will go briefly because these are all very complex mechanisms. So what are the fields we can tap this technology, we can use this technology? For gene editing in the fishery science and gene editing in monosex populations. Sometimes one sex of organisms will be growing faster, the other sex of gender is growing slowly. You can make them synchronized, and then gene editing in sterility of the fish, you can make sterile. You can make sterile fishes. For example, some predator fishes or overgrowing unnecessary fishes, you can make them sterile. So you can induce sterility. And then gene editing in the reproduction. If there are any reproductive disorders, you can make the reproduction synchronized, hormones secreted in normal way you can make. And then gene editing in fast growing fishes like cold water fishes, hot water fishes. You can make them grow in different stressed conditions. And then gene editing in ornamental fishes. These are all various applications, various areas where we can use this CRISPR-Cas technology. In gene editing in pigmentation, and then gene editing in the growth related problems, to address growth related problems, and then gene editing in the body configuration, gene editing in the oomycetes, and then okay, this is one small example. I am showing here the mechanism of CRISPR-Cas9 system used in the zebrafish. This was a recent study in the zebrafish and also in the tilapia. In the tilapia and zebrafish, a lot of CRISPR-Cas experiments were already done in the aquaculture area. Other fishes were still under trials. So this is the nuclease enzyme identifying the sequences with the help of this RNA. This is called guide RNA. This guide RNA is identifying the sequences and cutting the sequence of DNA on 2 double strand break, DSB, double strand breaks are induced. Okay, so far so good. It looks very attractive technology. So all technologies definitely used to have some side effects or drawbacks. So even though this is a fascinating technology, very recent and modern technology, it may have some problems, ethical issues, and some problems are ahead. We need to be very careful while using this CRISPR-Cas technology, because ethically the benefits must be greater than the risks. If risk is more, you should avoid such experiments. And the use of CRISPR-Cas9 technique can be risky and harmful since it may produce off-target mutations. So not only targeted mutations, sometimes off-target mutations may also be resulted. That is another dangerous sign. And this process is complex and it includes numerous ethical, bioethical issues and technical issues. Only experts can address these technologies. It will take still 1 or 2 decades to bring this technology to the field level. And this technique also lead to cell death or alteration of the cells through the cutting of unintended sequences, creating mutations that creates a lot of problems. So this is about CRISPR-Cas technology. Thank you.
I thank Dr. Mattareddy sir for his insightful regarding use of CRISPR's gene technology in aquaculture and its introduction like genome editing, which is the use of various technologies to make permanent gene changes in the genome in DNA. And also about genomic editing nucleases like talents, ZFNs and CRISPR genes and their specialties and importance in CRISPR repeats in genetics, which shows difference in individuals from one another, and a history of CRISPR gene which involves identification in E. coli in 1987, and working mechanism of CRISPR in simplified manner and specific definitions of protospacer, spacer, protospacer adjacent motif, which was abbreviated as PAM. CRISPR-Cas9 gene editing process, which involves in fishes like zebrafishes and tilapia. So, and also its applications and outcomes after genomic DNA cleavage. Thank you, sir, for sharing a valuable presentation to our audience. Thank you, sir.
ladies and gentlemen, esteemed colleagues, and honored guests. It is with great pleasure that I stand before you today to introduce our distinguished resource person, Professor G. Simhachalam Garu, stands as a distinguished figure in the realm of zoology, currently holding the esteemed position of professor in the Department of Zoology at Acharya Nagarjuna University, Guntur. With a stellar academic background and a profound dedication to the advancing scientific knowledge, Dr. Simhachalam Garu is a prominent scholar whose contributions have left an indelible mark on the field. Armed with the advanced degrees in the zoology, Dr. Simhachalam sir has cultivated a multifaceted expertise that spans various facets of the disciplines, such as discovering 5 new species and acted as a coordinator for 2 research projects. Besides, he acted as editorial member, Board of Intermediate Zoology book for NEET students, acted as a DSIR assistant coordinator, and evaluated several projects. He is also a life member of Indian Journal of Comparative Animal Physiology. His research endeavors have delved into the areas such as wildlife conservation, ecology, and biodiversity, reflecting a commitment to understanding, preserving the intricate dynamics of the natural world. As an educator, Dr. G. Simhachalam Garu has played a pivotal role in shaping the academic landscape of the Department of Zoology at Acharya Nagarjuna University. His teaching philosophy integrates theoretical knowledge with practical applications, fostering a holistic understanding of zoological principles among his students. Many of his mentees have gone on to the making significant contributions to the scientific community, a testament to his impactful mentorship. Dr. Simhachalam sir scholarly pursuits extend beyond the confines of the classroom. He has actively engaged in collaborative research projects, presenting his findings at 51 national and 19 international conferences. His research output, marked by numerous publications such as 14 national and 25 international articles in reputed journals, attests to the depth and breadth of his contribution to zoological knowledge. Please join me in extending a warm welcome to Dr. G. Simhachalam Garu to the National Seminar on Aquaculture Farming Advances, Challenges, and Opportunities. We anticipate that his wealth of experience and insights will enrich the discourse on contemporary issues in aquaculture farming. His participation promises to be a catalyst for intellectual exchange, fostering a collaborative environment that stimulates advancements in our understanding of the animal kingdom. So with this, I'm handing over the session to Dr. Simhachalam sir.
Dear students, as well as faculty members of college and my colleagues. Basically, I am a farmer and a professor. Farmer and a professor, farmer as aquaculturist. I have my own ponds. Some of the issues practically I observed at the field level. Basically, I am the taxonomist, and one point she forgotten, I discovered new species also. New species which documented in National History Museum, London. Sustainable aquaculture. In the aquaculture, why the India is second in position? Because of India is a peninsular India. Peninsular India means we are all well about the meaning of peninsular India. It is one of the criteria for we are the head in the aquaculture field. And the second one is the India is having different riverine systems, also perennial rivers. Okay, the third one is the coastline and the perennial rivers which facilitate for the aquaculture of in Indian subcontinent. Okay, because of these habitats, the farmers which constructed different ponds which they can farming aquaculture. Okay, in the basically the aquaculture means farming of aquaculture organisms which surviving in the habitats or water bodies. Okay, come under finfish, shellfish, and other crustaceans, molluscs, and aquatic culture of aquatic plants is also come under the aquaculture. Okay, see, already I told you that the vast coastline, riverine system, and ponds, which is the major causes for facilitate our aquaculture. Okay, the India is second in position after China. See, now we are producing 18.16 million tons of aqua products. This is only production by our farmers, not for the capture fisheries. Plus, the capture fisheries is also play a vital role because we have the vast coastline, vast coastline. Okay, this graphical representation which said that how the aquaculture, which is the increasing day by day, where we are started the production, and where now we are in 18.16 billion tons of million tons of aqua products we are exporting from to the other countries. Need for raising of aqua production. Why we improve our aquaculture? Okay, we know that our population, the figures which more than 1 crore 130 crores, more than 130 crores. Okay, for demand is more by the people, but this production is not increasing. Production is not increasing with like the people's demand. Okay, it is important for protein and elements and other byproducts for the human being. Okay, so that they are mainly depend on the fish consume for their one of the main food item. Okay, and also one of the important in the aquaculture sector is it added 1.5 trillion Indian rupees to our gross value. Gross value means including the agricultural and everything come under this figures. Support the entrepreneurs and support to the recent generations. Okay, this is important. Entrepreneur, this is the place, the industry which provide the opportunity for entrepreneurs. Already several entrepreneurs established their industries in terms of feed or maybe processing units processing units and hatcheries, etc. and etc. Okay, but if a student of aquaculture or a student of having the knowledge regarding to the aquaculture, we are also eligible to establish the or to convert entrepreneur in the particular aquaculture. Okay, support to the recent generations. Okay, it is give us the job opportunities. Job opportunity. Our students, which is what is that doing job in different companies, and they are offer more than 40, 60, up to 1 lakh or plus also some of the students which is drawing the salaries particular from the aquaculture industries. Okay. In earlier, before 1950 or 1970, okay, the people which is having less awareness towards the culture practices. They collect the seed from the natural habitats. Natural habitats. It is somewhat tedious to collect seed from the natural habitat for their demand, and fish stocking and rearing in earthen ponds. Okay, they completely depend on the earthen ponds in their own fields. Okay, water quality. Okay, the farmers which earlier practicing, okay, they are not much aware for maintaining the water quality parameters, and they used traditional feeds, and disease control is also somewhat difficult them by the farmers. Harvesting methods and marketing strategies are also very less, and managing practice also somewhat they are having with meager knowledge. Okay, so that in the earlier the farmers or the aquaculturists get good returns, but some of the farmers which is not having the good returns on par with the who practicing some technical knowledge. Okay, if this aquaculture, not only earlier, today also we are practicing traditional methods due to that operating or practicing using a traditional method. Okay, loss of natural habitats. For example, a best example here I quoted, how the natural habitats which is lost. For example, the best example for the our Ramsar site, Lake Kolleru. This lake earlier, this Lake Kolleru, which it is occupied by the several people after their selfish interest. Okay, the total lake which is disturbed and occupied and farming by the some of the entrepreneurs, and loss of genetic diversity and brood stocking also we can expect, particularly in the aquaculture. Water usage. For example, you see today it is one of the big problem for the in the aquaculture. Water usage. They using repeatedly using by one another. Okay, this the water which is released from the outlet, it is a inlet water of the others. Okay, this outlet water which is having contaminations. That is almost polluted water. Such a polluted water without treatment used by the other farmers. It is a big problem which we are observed in the present scenario. Okay. Intake of screening, without treatment, without purification, the same water which used by the other farmers. Okay, pollution and eutrophication, diseases and other things also which face the farmers which is facing in the aquaculture. Okay. Now, if you are compared to the earlier farming system and present farming system, the farmer cannot aware the technology, and within their knowledge they are operating their farms. Okay, it is one of the factor for the sustainable development of the aquaculture. We know that the meaning of sustainable development. Sustainable development means to use the resource for the present generation, for the development of the present generation without disturbing the economics or environment and social disturbances. But today the industry, the fishery industry or aquaculture industry, which is focusing only on the economics, but they are ignoring the other problems, environmental problems as well as community development. Only focusing the development of the individual, not for the others, and totally they ignore the environmental pollution also. When we balancing these three, environment, economics and social development, then only we can approach the sustainable aquaculture. Okay, for this definitely the farmers, the aquaculturists need to switch over for the need to operate the present technology and switch over from traditional methods to the other scientific methods. Then only our environment will protect and economically the aquaculture system, farming is viable. Okay, so from the beginning the farmers approaching extensive and semi-intensive methods. These are somewhat away from the pollution also. Environmental pollution also, because they stocking low densities and avoid supplementary feeds. Okay, completely the system or the culture which depend on the natural feed. Okay, no aerators and no labor cost. Okay, ఆ రోజున వచ్చినటువంటి లాభాలు అవన్నీ కూడా దానికి వాళ్ళకి ప్రాఫిటబుల్గా ఉండే ప్లస్ ద environmental pollution, the system is away from the environmental pollution also. Okay, semi-intensive, then to increasing the greed about the money, the farmers, the aquaculture is gradually switch over to the other methods in such a semi-intensive and intensive culture systems. When the farmer approached the farming system, the farming continuing using the intensive culture automatically increasing the problems like pollution and other problems also. Okay, for this intensive culture, what the farmers doing, high stocking density in a small area. It is one of the big problem. Okay, once we stocking more number of prawn or more number of carp species, automatically to increase the pressure on them. Okay, once increasing pressure on them to increase the bacteria, disease cause bacteria, and they face the diseases. Automatically they face the diseases. Okay, once if increasing the diseases, automatically the farmer using the antibiotics and other chemicals and drugs. This is the big problem which the industry which is facing. Okay. Okay. Then to avoid the problems which arising from the intensive culture, okay, to maintain intensive culture, but you switch over to the method, from the traditional method to the scientific methods. Okay, in the scientific methods, the farmer which maintaining high densities using technology, okay, some extent the farmer is avoiding the diseases, pollution, as well as the other problems. At the same time, the farmer getting more money also. Okay, the money also. Okay, cage culture. This is one of the best method for meet the demand of the people. Already I told you that we have a good number of natural resources like rivers, sea, reservoirs and canals and natural lakes. These are all is important places which to operate cage culture without affecting without pollution of the habitat. Okay, and also the farmer or the aquaculturist cannot lose money also. Okay, in the cages sometimes they are fixed cages and floating cages. There also the farmer which used the supplementary feed some extent, but the fish or prawn which can take the natural feed directly from the surrounding environment. It is one of the main advantage for the intensive culture and intensive culture of a cage system. Okay, cages are made with different nets. These nets which can stop the escaping of the species from the main area. and benefits. These are important for if we culture using the cage system. Natural feed. For using natural feed, if the farmer which can get some advantages to reduce the feed cost, one thing, and low investment. Okay, single investment. If he invest for construction of the cage only. Several years, the same cage is used for the culture of the carps or what the desired species which he can choose for culture in a cage system. Okay, reduced pollution, no pollution or maybe reduced pollution, because they avoid the supplementary feed. They can take the feed from their surrounding environment, and harvest is also very, very easy, because they are all concentrated in a particular cage. It is one of the what is that advantage, economically advantage for the farmer. Okay, we are all aware how difficult today the harvest of a acre or 2 acre pond. It is what is that connected with the labor. It is much cost for harvesting in the traditional method. Control predators and competitors. No predators and no competitors in this particular cage culture, and high productivity with limited area. Okay, what is the actual greed of farmers to the criteria is money. Okay, that also they can meet particular in the intensive culture. Okay, relocation of cages, resource used efficiently. All resources, all our natural resources. For what? Our natural resources. Natural resource is a property of people, not for a individual. We have to right to culture in our natural resources after a little bit of effort or after permission from the local bodies or maybe the local government. So here also we can maintain the sustainable development. Okay, raft culture, it is also one of the culture like cage culture. This is also, okay, what is that to reduce the pollution and everything. Okay, here in the raft culture, the concept is that the ropes which is hanging in a fixed logs, okay, the spat or the juveniles or embryos which is attached to the ropes, and they can take the feed from the surrounding area, diatoms or whatever they adopt to feed, they can eat from their surrounding environment. This is here also we can avoid the avoid the pollution and economically it is also viable practice. Okay, particularly by this culture is suitable for the oyster, mussels and other crustacean and molluscan organisms. This method is not suitable for the prawn as well as the carps. Okay, raceway culture, it is one of the scientific method to avoiding the problems which what we are facing. The most important or the problem is pollution of the habitats, Okay, this in the raceway culture, the drain pipes which is fixed in the bottom of the pond. Through that the drained, and it is again it is filtered, again it recirculated and sent into the main system. Here they won't use aerators, and here also avoiding the diseases and everything. Okay, any type of fish which is it is suitable, particular in the raceway culture. Okay, RAS circulation, recirculatory aquaculture. Recirculatory aquaculture. The problem is that the farmers or the other people is unaware for the different methods which existing in the aquaculture. The RAS system is very, very important and profitable, as well as the good farming system in the present scenario. It is occupying the second place after the bioflak method. Okay, here you see in sustainable development, use resource effectively. The main aim is that use resource effectively. Okay, here for aquaculture, we are using the land, more and more land. Okay, for this system, not much land is not necessary. This is the one important aspect which we observed in the RAS system. A small places which we can meet the some extent it can serve this approach which can serve the farmers greed also. అతను ఏంటంటే బాగా డబ్బు సంపాదించాలి అనేది ఒక ఆలోచన, అంతే కదా. ఓకే, ఇఫ్ యు ఆర్ అప్రోచ్ దిస్ సిస్టమ్ డెఫినెట్లీ హీ కెన్ రీచ్ ది గోల్ without damage of the our environment. Okay, ఒక 1000 or 500 acres లో పెంచేటటువంటి చేపల్ని if you are going to culture or adopt to system 1 or 1.5 acre lo, the same farmer is able to take the much production. అంటే 500 ఎకరాల్లో వచ్చేది ఆటోమేటిక్గా we can able to take if you are operating this RAS system. Okay, and it is also operate in the multi-stored buildings also. A little bit of land is enough for the RAS system, and also a little amount of water is enough for RAS system. RAS means recirculatory aquaculture. Use the water and filter, chemical filtration or maybe the filtration, whatever you method you adopt. Okay, the same water is using again and again. Okay, there we avoid the water problem also. We are avoiding the land problem also. If you have the 10 cents of your homeland, 10 cents or 5 cents, there also we able to construct the RAS system. ఎవరైనా సరే వితౌట్ నాకు పొలం ఉంది, పొలం లేదు, నాకు 2 సెంట్లే ఉంది, 5 సెంట్లే ఉంది, 10 సెంట్లే ఉంది అని కాదు. ఎవరైనా సరే if you are interested, we are able to operate the RAS system, and also here we are maintaining the anything like the high densities also. Okay. Okay, bioflak method. Okay, after RAS system, this new method which is introduced recently, several farmers which adopting this bioflak method also. Okay, like RAS system, it is also, what is that, to construct in the multi-stored buildings or within a stipulated area, within the small area. Okay, here to reduce the feed cost, and also to reduce the contaminations, pollution as well as the other contaminations, to avoid the disease also. Okay, here in the method, what the excretory materials which is released from the fish, it is a big problem if they are retained in the long period. Okay, scientifically, what our scientists which experimentally improved this method, this excretory materials which is converted into bioflak after using the carbon source or other microorganisms. This bioflak which containing the microorganisms as well as bio, and directly the material which eaten by the fish or prawn, without what is that, recirculating the water. Here reduce the feed cost, and also available high protein to the consumer also. High protein to the consumer. Okay, this method also we are observing in several places in and around our coastal area. Okay, what my suggestion in aquaculture, this RAS system as well as the bioflak method is not necessary for the a big farmlands. మనకి పెద్ద ఏం అక్కర్లేదు. ఒక ఏకరు లేకపోతే అరెకరు మీకు ఉన్నది ఏదైనా, లేకపోతే మీ ఇంటి దగ్గరే మనం వీఆర్ go for the construction of these methods. Okay, to achieve the goals of sustainability, monosex culture, now we observing this particular culture is operating by only one species, tilapia. Okay, why not our scientists, including myself and whatever the intellectuals, to develop that technique in other species. For example, in the carp culture, we are culturing male and female. While in harvesting, particularly in the summer, okay, if they are having the half kg or more than the eggs in their stomach, okay, the period is not suitable for the harvesting, because to reduce the cost of the fish. If we improve the monosex culture, particularly in the carps also, it is viable, and the farmer which can get the good returns. Okay, biological feeds, it is somewhat difficult to improve the biological feeds, because biological feeds means cultures, rotifers or moina, culture of moina, copepods and other species which the fish which can prefer these species. Okay, naturally to improve their density is somewhat difficult. Okay, and formulations of the vaccines, this is a big problem for our industry. Why the institutes which is located in the worldwide or in India, why they are not concentrated for the development of the vaccines? Okay, recently, how fast the scientists which developed the vaccines for the COVID-19, why not they are not concentrating vaccines for the aquaculture industry? This is one of the big drawback for our aquaculture. Okay, for development of vaccines, necessary support from the government, state government as well as central government. Here big problem is they totally ignoring the aquaculture system. Okay, because of our governmental activities or policies, our aquaculture which cannot sustainable and facing different problems. They are rising the current charges and they are allowing indiscriminate pond constructions. If it is very important, if you are giving a small amount of money from our secretariat level to the district fishery officer, except collector and sub-collector is not taking the money, but if you construct or digging a new pond, okay, taking money from the secretariat fisheries employee and irrigation employee and drainage employee and then the fisheries inspectors, mandal level, MRO and FDO, everybody which can to take the huge amounts because I know this practical problem. I am also victim for particular in this aspect. Okay, what I suggest that to allocate particular zones as aquatic zones. You allocate particular zones as aquatic zones, or else to educate the farmers in other methods from the traditional methods. Okay, then only the aquaculture which can sustain. ఎంత మంది ఫార్మర్స్ లాస్లు వచ్చి వాళ్ళు ప్రాబ్లమ్స్ ఫేస్ మనం అనుకున్న ఆక్వాకల్చర్ చాలా పెద్ద ఇండస్ట్రీ, వాళ్ళు చాలా డబ్బు ఎవరికంటే ఉన్న వాళ్ళకే వస్తుంది కానీ లేని వాడికి లేదు. ఎవరికైతే డబ్బులు ఉన్నాయో వాళ్ళే దే కెన్ ఎఫర్ట్ సంథింగ్ వాట్ దట్ టెక్నాలజీ and they are effort the employees who know the technology. They can achieving their goals, not for other people. Okay, finally, artificial intelligence is the it may be the solution for the problems which we addressing the earlier. Okay, artificial intelligence, now this technology is used in not only one place, everywhere. Okay, why not we using this technology in the aquaculture sector? Okay, already it is using somewhere and they can get the good results also. Okay, this artificial intelligence is easy to analyze once we provide a huge data and within a click it can solve the problems like water quality problems, disease problems, growth versus feed and other complications also. It can immediately give the solution for the farmers. Okay, one time if you are feeding through the algorithms, it can solve the several problems who facing problems facing by the farmers. Okay, for this the government effort is needed. The government effort is needed. Without the government support, for example, you see today in Andhra Pradesh or maybe the Telangana, other states, within a single click, without single click, the money is circulating in their footsteps. Why not the same technology is using in the aquaculture? Okay, we have a good number of employees. 2 lakhs employees are there in the village level. They are taking much amounts as salaries. Okay, using their services, you collect the data from the farmers. What type of problems they are facing? And to address the problems and use the artificial intelligence. For example, you see, మనం చాలా సార్లు చెరువు దగ్గర పలానా అబ్బాయి చనిపోయాడు, షాక్ కొట్టాను అంటాం కదా, షాక్ కొట్టి చనిపోయాడు అని అంటాం. Why not to provide the robotic, why not use the robotic technology for the farm, in the farm side? Okay, మేత జల్లాలి అని అంటే, మేత వేయాలనంటే వీళ్ళు కావాలి, దానికి కూడా రోబోట్ టెక్నాలజీ యూజ్ చేయొచ్చు. బట్ ద ప్రమోషన్ ఈజ్ ద ఇంపార్టెంట్. ద గవర్నమెంటల్ సైడ్ it is important to encourage this type of activities. Okay, disease control, feeding management, water parameters, efficient feed formulations, growth rates, minimize environmental impact, early detection of disease, parasites, abnormalities, everything which we can solve the under AI technology. Okay, if we are effectively manage with sustainable aquaculture practicing, only when we use the integration AI, artificial intelligence, as well as BMPS, better management practices. If you combine both things, okay, the farmer or the state or country which can lead, definitely we are in the first place soon if you are operating all these things. See, we have the several institutions. Their basic aim and basic work is to develop such things. What we discussing just now, all such problems which is definitely or must to address by these people, NFD, ICAR, CMFRI, whatever, all these institutions which the employees or the scientists which recruited only for to solve the problems of aquaculture. Okay, so far you great or whether it is great or not, so far only we are to develop new species, one species, and second species is not new species. Our scientists are investigated and they suggested the species is suitable for culture in a freshwater is a Lithopenes venami. Okay, and the other species which it is promoted by our scientists is the Jayanti Rohu. Jayanti Rohu is only the new species so far our scientists developed. Jayanti Rohu, except no. Every species, Katla or maybe the Rohu or other things, other species which we depend the natural habitats. Natural habitat. That is our greatness of our things. Okay, the main problem is the government. The main problem is the governments. To be frank, the main problem is the government, and also greed of the human beings. Okay, thank you.
So please, sir. So please, sir. Now I invite Dr. P.V.V. Satyanarayana. Now I invite Dr. P.V.V. Satyanarayana Garu, lecturer in zoology from Sri Y.N. College, Narsapur, to give keynote address on this talk. So please.
Good afternoon. I am very much thankful to Dr. G. Simhachalam Garu, who enlightened his